Hello students, the topic for today's lecture is Pheromones Part 2. In the first video, I had discussed about pheromones, how they were discovered and how important they are in the communication in animal world. Today we will be discussing effect of pheromones. A pheromone affects the behavior in two ways. One effect is known as releaser effect and another is known as primer effect. Releaser effect is that which affects as soon as the pheromone is released. When the pheromone is released by the animal, it stimulates central nervous system of the recipient or another animal and it produces an immediate change, then it is called as releaser effect. But if it takes time in changing the behavior of the recipient, then it is called as primer effect. So, in the primer effect, pheromone is secreted and that secreted pheromone, it alters the set of physiological reactions in the recipient and this physiological activity in turn affects the behavioral pattern of the recipient and later by an appropriate stimulus that behavioral pattern is released. So this effect is called as primer effect. So releaser effect is immediate and primer effect is delayed. We can also call it as delayed effect of pheromones. Now suppose we will discuss both the effects with examples. Suppose a pheromone is released by animal A and it is perceived by animal B and it is acting on central nervous system of the B and it releases immediate behavioral change, then this kind of quick action is called as releaser effect. For example, it has been seen if we crush one snail by the side of the river and then we will see that immediately so many snails which are there, they will all go inside the water. Why? Because that crushed cells are emitting a fear pheromone which is being perceived by other snails and they sense that there is danger, so they will immediately go inside the water. Similarly, termites, we all know that termites are social insects and they live in a colony. If termites discover a breach or a crack in their mound, then soldier termites or whichever termite discovers that breach will emit an alarm scent that will bring worker termites to work and they will immediately start repairing the crack. So these both are examples of releaser effect. Pheromone secrete हुआ, एनिमल ने दूसरे एनिमल ने ऑफ द सेम स्पीशीज उसे परसीव करा, रिसीव करा और उसके बिहेवियर में इमीडिएट चेंज हुआ. दिस इज कॉल्ड एज रिलीजर इफेक्ट. एंड pheromones that produce releaser effect are widely spread in animal kingdom. Sex attractants which are found in insects, they also fall under the category of this releaser effect. जैसे in the last lecture we had discussed about trail of ants, वो भी एक consequence है, example है of releaser effect. Similarly, if we crush a bee, जैसे हमने snail की बात करी, if we crush a bee, then immediately you will be attacked by hundreds of bees, क्योंकि that crushed bee, by crushing a bee, an alarm pheromone is released from its body and it is perceived by other bees which are around only and they will start attacking the person. Now let us take examples of primer effect. In primer effect, a pheromone is released by the animal A. It is being perceived by animal B. It is acting on central nervous system of B and is bringing change not immediately in the behavior, but it is bringing change in the physiology of the receiver or the recipient, and that physiological change is being released by an appropriate stimulus and then behavior will change. So it is a delayed effect of pheromone. इस इफेक्ट के एग्जाम्पल्स कम हैं बट दे आर वेरी इंटरेस्टिंग. In 1955, several mammalian endocrinologists like Lee and Boot, they discovered such type of pheromones in female mouse. What they observed was that if large number of female mice are left in a cage together, then their estrous cycles get synchronized. So this is an example of delayed effect. They further observed that if female mice are kept away from males for a long time, then their estrous cycle is stopped completely. So these two are examples of delayed effect कि ये असर होता है there is a change in the behavior, physiological behavior of female mouse, but it takes some time. एंड स्टॉपेज ऑफ ईस्ट्रस साइकिल जो हमने सेकेंड एग्जाम्पल लिया कि ईस्ट्रस साइकिल ऑफ फीमेल्स इज स्टॉप्ड कंप्लीटली इफ नो मेल माउस इज देयर इन देयर विसिनिटी. वाई? बिकॉज a pheromone or a scent is emitted by male mouse that has capacity to initiate the estrous cycle of female mouse. So in the absence of that pheromone, the estrous cycle of female mouse is not initiated. So scientists came to the conclusion that pheromones secreted by male mice are perceived by female mice and they gradually affect the reproductive physiology of female mice, and consequently there is a change in the reproductive behavior of female mice. So this is the example of delayed effect. What we said in the definition was that the pheromone is secreted, it is perceived by central nervous system, then it changes, it brings about some change in the physiology of the recipient, and then by the stimulus of that there is a change in the behavior. Lee and Boot further investigated that when female mice are kept in groups, there was an increase in the number of pseudo pregnancies, and this could be prevented by removing olfactory bulbs or by isolating the females, and they called it Lee-Boot effect. That means by removing the olfactory bulbs, females cannot perceive or cannot sense the pheromones of one another. So again this is an example of delayed effect. One more example of this was studied by Helen Bruce in 1961. She observed that odor of strange male mouse, it blocked the pregnancy of the newly conceived female mouse, whereas she observed that the odor of the original male mouse did not disturb the pregnancy, whereas the odor of the strange male mouse suppressed the secretion of the hormone prolactin, which is responsible for continuing the pregnancy, and as soon as the secretion of prolactin was suppressed, then normal estrous cycle was restored. This is known as Helen Bruce effect. So Lee-Boot effect and Helen Bruce effect are both examples of delayed effect of pheromones or releaser effect. Now other examples of primer effects, they come from social insects like honeybees, termites and ants, and they all have been widely studied for this purpose. So among social insects, members of the same colony, they share same pheromone, and their pheromone differs from the pheromone of the members of other colonies of the same species. अगर हम बीज की बात करें तो क्वीन बी प्रिजर्व्स हर मोनार्की बाय producing a pheromone that is called as queen bee pheromone, and that pheromone, it inhibits worker bees from making new royal chambers. उसके इस pheromone से worker bees को पता रहता है कि queen is alive and we do not need to construct new royal chambers. Then termites में भी देखा गया है कि caste production is regulated by number of individuals of the caste that are already present. All these examples involve delayed primer effect and knowledge of pheromones in the animal kingdom is well known since 1970s. और इन pheromones को जब इनकी knowledge हुई तो manufacturers are marketing them for pest control, and these pheromones are also used to attract or to repel animals and bugs to prevent crop damage. So this is a very important use of insect pheromones. Now this slide shows glands producing pheromones in insects. These are called as scent patches. Wings, they are present in the wings. Then these are scent glands, mandibular glands. These are present in the anal glands. So these are various types of scent producing glands or pheromone producing glands in insects. Now structures receiving pheromones. We have discussed just now that there are various glands which produce pheromones. Similarly, there are various structures which receive pheromones in insects and other animals. Now Swiss entomologist Forel, he found that various structures are present on ants' antenna and other insects, and they receive pheromones. These structures were flat in some insects, cup shaped in some others, and usually they are in the form of short stubby hair. And all these structures which are perceiving pheromones are supplied by nerves. And when pheromones are released by conspecifics, they are perceived by these specialized structures and messages are sent to central nervous system. And although there are variations, सब में structures perceive करने वाले थोड़े से different होते हैं, but all share the basic plan कि there is a receptor cell and a supply of nerves. And some animals have got antennae specially designed to receive pheromones. Most of the animals have got antennae and they receive pheromones. Now functions of pheromones. Pheromones perform a variety of functions in different animals. Pheromones are used as, or they act as sex attractants, and they in insects also, in higher animals also, they bring the sexes together for mating. Therefore they are also called as sex attractants. Social insects के जो pheromones होते हैं, they are required for, or they are utilized for communication between the conspecifics, and they are also responsible or very essential for the maintenance of the colony structure or caste system. Now, as we said that pheromones are used for scent marking also. Vertebrates use pheromones for scent marking. Now, many vertebrates, they produce pheromones which have got strong odor or scent, and these odors, they carry out many important functions. Mammals mark their territories, mates, pups, their habitat, etc. with their pheromone which has strong odor. And this odor or scent is released in many ways in vertebrates. It can be released along with urine, feces and saliva, and they are also produced by special glands in these animals. Thank you.